How's it going, my fellow nerds? In this video presentation, we shall be talking about glycolysis. So what is glycolysis, for those of you who may be wondering? Glycolysis is a metabolic pathway that takes place in the cytoplasm of the cell and converts glucose into pyruvic acid. So how does it do this? Well, glycolysis begins with a 6-carbon sugar called glucose and starts in a stage called glucose priming. In this stage, the glucose molecule is so stable that it has to undergo a set of reactions to become phosphorylated so that it can be reduced and ultimately produce energy. To be more specific, in this stage, the glucose molecule is phosphorylated by a kinase protein in order to decrease the sugar stability. This initial phosphorylation, done by a hexokinase, creates a glucose 6-phosphate, and does so by stripping a phosphorus atom from an ATP molecule in the cytoplasm and attaching it to the glucose. This glucose 6-phosphate is then altered by another enzyme, which makes it even more unstable by altering its form, turning the 6-carbon glucose into a 6-carbon fructose, or a fructose 6-phosphate. The fructose molecule that's produced will be phosphorylated once again by another kinase enzyme to make a fructose 1,6-phosphate, which is even more unstable than its previous forms. This molecule is unable to retain its form for very long and will ultimately split itself into 2 separate 3-carbon sugars that are far more stable. These 3-carbon sugars are energy-rich G3P molecules, which enter the next phase of glycolysis, which is called the energy harvest. In energy harvest, each G3P molecule is reduced by a phosphate from the cytoplasm, as well as an electron carrier, an NAD+, to form an NADH electron carrier molecule and a single molecule of glyceric acid. This glyceric acid is a 1,3-diphosphate glyceric acid. The glyceric acid then gives up a phosphorus atom to an ADP molecule, or an adenosine diphosphate, and creates an ATP molecule, which can then be used to meet the cell's energy requirements. The molecule then undergoes multiple stages of isometric rearrangement, where the phosphorus, which initially starts on the far end of the carbon chain, shifts into the position towards the center of the carbon chain. This is done by a set of highly specific enzymes in a multi-step process. When the phosphate enters its correct position, it forms a double bond with the number 2 carbon. This forms phosphoenolpyruvic acid, or PEP, or PEP. This PEP molecule gives up its last phosphate to another ADP molecule when catalyzed by another kinase protein. This produces another ATP molecule as well as a molecule of pyruvic acid. Because 2 G3P molecules are produced from a single glucose molecule, this process of energy harvest is actually happening twice, one for each G3P, per glucose. So what exactly just happened? In summary, glycolysis utilizes 2 ATP to phosphorylize the initial glucose molecule, but in doing so ultimately produces 4 ATP molecules as well as 2 NADH carriers and 2 pyruvic acids. This means that we have a total net gain of 2 ATP. The pyruvic acid that we made will make its way to the Krebs cycle, and the NADH molecules will be utilized to synthesize yet even more ATP in the electron transport chain. So why is glycolysis so important? Glycolysis is the first step in energy production for every living thing, except for a very small amount of extremophile archaea which live near oceanic sulfuric vents. If that wasn't cool enough, here's another fun example. So let's compare a bacteria and a cat, because this couldn't be an awesome video without a cat, right? Both organisms require glycolysis to meet their energy needs, but being so simple and requiring less energy, the bacteria only requires glycolysis to meet its energy requirements, while the cat has to undergo the Krebs cycle as well as the electron transport chain to meet its energy needs. This means that the cat requires oxygen and more glucose molecules in order to produce more energy to sustain itself, while the bacteria does not require oxygen and utilizes less energy. This means that the bacteria is more resilient because it's less likely to be affected by atmospheric conditions or the amount of available energy. So, in this case, the bacteria wins. Sorry, cat. This video was brought to you by the Reynolds High School Biology Department. Thanks for watching.